Good day, dear students. We are beginning our lecture. Today our lecture is devoted to the blood proteins, immunoglobulins and hybridoma technology. Let's begin. I shall try to say everything slow, that you will have possibility to write after me. Please write the topic: blood proteins, immunoglobulins. There are many different proteins in our blood which fulfill different functions. If we shall divide proteins with the help of electrophoresis, we can receive some protein fractions. The quickest one is albumin. Then we have alpha 1, alpha 2, beta and gamma globulins. The largest fractions of blood proteins are albumins and immunoglobulins. Last are situated in gamma globulin fractions. And immunoglobulins are the main object of the research, of scientific research of our department of biochemistry for many years. We are specialists in this field, and I want to tell you some interesting moments devoted to the structure, functions of immunoglobulins, and to hybridoma technology, which is the process of obtaining of large amounts of immunoglobulins with desired specificity. Let's begin. On the picture, you see the structure of the molecule of immunoglobulin G. On this example, we shall study the structure of all immunoglobulins. You can see that this is a very large molecule. Each small ball, which are shown here by red, blue, light blue colors, are amino acids. The molecule of immunoglobulins has very many amino acids, and the molecular mass of the whole molecule is 150,000. So it is a huge molecule, very large molecule. 150,000. This molecule consists of 4 different chains. 2 heavy chains, one is light blue, second is dark blue, and 2 light chains. Red, this is on the left side, and second on the right side. Don't write this information now, because later I shall show you more simple scheme of the structure of immunoglobulin, and you will copy it in your notebooks. Before I shall change the picture, I want to show you also these orange balls that show carbohydrate component of immunoglobulin. The conclusion is that immunoglobulin is a complex protein, because it has not only amino acids but also carbohydrate component, this one, which is situated between 2 heavy chains. Now, very short history of the study of immunoglobulins. The structure of immunoglobulins was discovered by 2 scientists. One of them is Gerald Edelman. Now you see his portrait, who worked in New York in United States of America, and the second one was Rodney Porter, He's from Great Britain, and he worked in London. These two scientists studied immunoglobulins for many years, beginning from 1959, and they divided the molecule of immunoglobulins into fragments, into chains. Then they studied sequence of amino acid sequence of immunoglobulins, and finally they produced the scheme of structure of immunoglobulins, which is completely correct. This was later confirmed by many methods, including, for example, electronic microscopy. In 1972, these two scientists received the Nobel Prize for their investigations of immunoglobulins. I want to repeat that they worked separately. One had his laboratory in the United States and second in Great Britain, but they studied the same object, and finally they published very important papers on the structure of immunoglobulins and received Nobel Prize for these investigations. Let's now study the structure of molecule of immunoglobulin. This is an example of immunoglobulin G, and I ask you to copy this figure to your notebooks. I shall say slowly, show you all details, and you will have enough time to copy this picture. If you have different colors, blue, red, please do it with different colors. If not, somehow show the difference in different pieces of the molecule. As I told you, and now I am repeating and you are writing, that the molecule of immunoglobulin G consists of 4 polypeptide chains. 4 polypeptide chains. 2 chains are heavy. This is one heavy chain. This is second heavy chain. And 2 light chains. One and second. You can see from this picture that the molecule is completely symmetric, and left side is equal to the right side. As I told you, the whole molecular mass of the molecule of immunoglobulin G from human blood is 150 thousandths, and you can simply calculate now the molecular mass of heavy chains and light chains. Look, every chain, at first we must explain, every chain consists of the repeating parts, which we call domains. So light chain has 1, 2 domains, 1, 2 domains, and the heavy chain has 1, 2, 3, 4 domains. Don't look at the blue part, we shall tell about it a little bit later. So we have in the whole molecule of immunoglobulin 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 domains. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So if you divide 150 thousandths to 12, you will receive the molecular mass on one domain, and then you can simply calculate the molecular mass of heavy chain and light chain. Heavy chain 50 thousandths, second 50 thousandths, and light chain 25 thousandths and 25 thousandths. So in the whole we have 150 thousandths. Different chains in the molecule are linked with such lines, and these lines show the disulfide bonds. So these are covalent disulfide bonds between different chains. There are also such disulfide bonds inside of each domain. On other pictures, which I shall show you a little bit later, you will see the position of these disulfide bonds inside of the domains. Some domains have very stable primary structure, and we call them constant domains. Constant domains, because in different molecules of immunoglobulins this structure is equal. Constant domains are shown on this picture by black color. So you can see that there is one constant domain in light chain and 1, 2, 3 constant domains in the heavy chain. How to call them? Very simple. We write large letter C, and then if together with C we are writing index as large letter L, it means constant domain of light chain. In the heavy chain we have same system of letters, but as we have 1, 2, 3 different constant domains, we add also the number CH1, CH2 and CH3. What does it mean? Constant domain of heavy chain number 1, or the first constant domain of heavy chain. Then constant domain of heavy chain number 2, and we can say the second constant domain of heavy chain, and also constant domain of heavy chain number 3, or third constant domain of heavy chain. But some domains are very different in different molecules of immunoglobulins, and as a result we call them variable domains. There is one variable domain in heavy chain and one variable domain in light chain, now we can explain what does it mean VH. VH is variable domain of heavy chain, and VL is variable domain of light chain. Why these two domains here and two domains here are very variable? Because there is an antigen binding site between these two domains, and you understand that antigen binding site in each immunoglobulin must be different, because different immunoglobulins can specifically react with different antigens, and this specificity depends on the structure of the variable domains. Now we can explain that immunoglobulins are antibodies. What is antibody? Antibody is the main molecule of the immune system of vertebrate animals and also of people. And antibodies are molecules which can mark foreign substances, which we call antigens. And after this interaction of immunoglobulin antibody with the antigen, our immune system is trying to kill antigen. And this is our defense from different sometimes dangerous antigens. And I repeat that antibody, immunoglobulin, react with antigen by specific structure of antigen binding site, which is situated between variable domains of heavy and light chain. Porter, I told you already about this scientist. Porter influenced on the molecule of immunoglobulin with the help of the enzyme papain, and this enzyme cut the molecule here where we have blue part, and this molecule was divided into three parts. This one, they are equal, and this one which is different. These two parts which are equal we call FAB fragments. What does it mean? F is fragment and AB is antigen binding fragment. You understand why we call this antigen binding fragment? Because these fragments still has antigen binding site, and this fragment still has possibility to interact specifically with antigen. The third part we call FC part. Why FC? There are two explanations. First is fragment constant. It is understandable because it consists of only constant domains, and second explanation is fragment crystalline. Why crystalline? Because this part of the molecule is hydrophobic, and in solution, in water solution, it can produce a sediment which looks like crystallic structure. It is not real crystal, but this is a sediment, and we call this fragment crystalline or fragment constant. But there is another explanation. As there are three fragments in the molecule, we can call them by the letters, first letters of the alphabet, A, B, C. And as fragment A and fragment B are equal, we say FAB, FAB, and third is of course FC. So there are many explanations of the names of these parts of immunoglobulins. So let's repeat: FAB fragment, fragment antigen binding, and FC fragment constant or fragment crystalline. Let's look now at this blue part of the immunoglobulin. This is the flexible part, and we call this part hinge region. Hinge region. What is hinge? Articulatio, if we shall say in Latin. Hinge region. This place where molecule is flexible. So these parts, FAB parts, can move up and down, because this is a hinge region. Except antigen binding site, which is situated here, as you remember, there are some other different active sites on the surface of the molecule of immunoglobulins, and one is shown here as complement binding site. As you can see, complement binding site is situated on the surface of CH2 domain, here and here. So we have two complement binding sites. What is complement? Complement is a system of immune system proteins which are also can be used by immune system to fight against foreign substances, antigens. when for example, when the molecule of immunoglobulin interact with antigen, for example, with the molecules on the surface of bacteria, after this, proteins of complement system, beginning from first component, interact with CH2 domain, and then we have activation of the system of complement, and they produce enzymes which and also hydrophobic structures which can destroy, for example, the membrane of the bacteria or membrane of the cell which attacks the human organism. So this is a very important place of the molecule of antibody. And as I told you, let's look once more the previous photo, previous figure. Between two heavy chains there are carbohydrate components, and let's look on our scheme. Yes, they are shown here, but this small quadrate, black quadrate, they are also interact with CH2 domain. It is shown here from the outside, but normally, as you have seen on the first drawing, this molecule is more complicated than this scheme, and this carbohydrate is situated between two CH2 domains. Different domains, for example, VL and VH, can interact one with another with hydrophobic bonds, as well as CH1 with Cl and CH3 with CH3. But CH2 cannot interact hydrophobically with another CH2 because of the presence of carbohydrates between these two domains, and we shall see this on one of the next pictures. If this figure is completely copied to your notebook, we can go on. Now I shall show you some another schemes of the structure of immunoglobulins, and you will now simply explain me what does mean different colors or different letters of on the next schemes. For example, this one. This is the same molecule of immunoglobulin G, but this scheme is more simple than previous one. Now I am asking you, and you are answering me, and then try to answer, and then I shall confirm your correct answers. What is shown on this picture with red color? Now you are answering, and now I am confirming. Red color shows light chains. Second question. What is shown by blue color? I think you answered correctly. Blue color shows heavy chains. One heavy chain, second heavy chain. This more thin part of heavy chain shows hinge region, flexible part. What is SS? This is understandable. This is disulfide bond between light chain and heavy chain, light chain and heavy chain, and between two heavy chains. Other things are written: antigen binding site, and heavy chain. And this is not written, but this is understandable. This is light chain. Let's go on. This is the scheme of the same molecule, but here we can see some important details. Look, each domain is formed by the large loop of the polypeptide chain, and this loop is fixed by disulfide bond. Now you can simply understand why it is necessary to have a disulfide bond in each domain, because these bonds fix the loop of domain. You can see such a bond in each domain, here, 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 and also in light chain. What is red? Red is here one light chain and here second light chain. What is blue? Blue is heavy chain and here second heavy chain, the second heavy chain. You can also see the letters that show the names of domains. Variable domain of heavy chain, first constant domain of heavy chain, second constant domain of heavy chain, and third constant domain of heavy chain. And we have variable domain of light chain and constant domain of light chain. What is situated here? You remember, here we have antigen binding site, and here we have second antigen binding site. Disulfide bonds here, here, and here are necessary for connecting different chains together in one molecule. Every polypeptide chain, as you very well know, has the beginning and the end. The beginning is the place where amino acid have free NH2 group, and C and it's the place end of the polypeptide chain when the last amino acid have simple and free carboxyl group. On this drawing you can see that the beginning of all polypeptide chains are situated in the beginning of variable domains. So this is N end of polypeptide chain, this is C end of heavy polypeptide chain. This is the same situation, and also in light chain we have N end here and C end here. So antigen binding sites are formed by N ends of polypeptide chains. This frame shows, I'm waiting, you are thinking, and now I'm saying, this frame is showing FAB fragment, second FAB fragment, and this frame shows FC fragment. Fragment antigen binding, fragment antigen binding, fragment crystalline or fragment constant. Let's go on. This is the same molecule of immunoglobulin G. But here we can see the secondary structure of immunoglobulins. Second, the main type, this please write, the main type of secondary structure of immunoglobulins is beta structure. The main type is beta structure. And parts of segments of beta structure, beta sheets, you can see by these arrows, red arrows and blue arrows. As you remember, blue color shows heavy chains and red color shows light chains. But here you can see that why hinge region is so flexible. Look, because in the hinge region there is no real domains, and these polypeptide chains allow to be the molecule flexible in this place. I repeat once more, and you must write this is very important, that the main type of secondary structure of immunoglobulins is beta structure. This was not clear for the long time, and the first man in the world who discovered this, who discovered that the main type of secondary structure of immunoglobulins is beta structure, was very famous scientist German Vasilievich Troitsky, professor who was the head of the Department of Biochemistry of our university for many years. He became the head of the department in 1953 and worked as professor in this department until his death in 1992. So he was head and professor of the department nearly 40 years. And he made many important discoveries about which our teachers told you during the first class, and we also discussed some of these discoveries in different lectures. And one of the most important discoveries was the discovery of the beta structure in immunoglobulins. You can see that this is not so important, but I shall tell you that before these important investigations of Professor Troitsky, all scientists thought that the main type of secondary structure of immunoglobulins was alpha helix. And when the paper by Troitsky was published, many scientists said that it is impossible. And it was a real battle, a real struggle between different groups of scientists against Troitsky, and these people said that what he thinks that he had published is nonsense. But later, by the X-ray analysis, this fact that the main type of structure of immunoglobulins, secondary structure, is beta structure, was completely confirmed. And Professor Troitsky was right, and please write that his important paper about this, about the main type of secondary structure of immunoglobulins, was published in 1965. In 1965, in the journal Biophysics. 1965 in the journal Biophysics. On the next picture, you can see the beta structure of immunoglobulins at larger magnification. My question to all of you, please tell me, what do we see on this figure? I hope you understood that this is one of the light chains of immunoglobulin, because here we have Cl domain and here you have Vl domain. And look, the polypeptide chain mainly forms sheets of beta structure, which are shown with these brown arrows here and also here. And believe me that all other domains of the immunoglobulin have the same structure. This was discovered by Professor Troitsky, who was head of the Department of Biochemistry of our university. He is my teacher, and I am very proud that this great scientist worked in our university. Let's go on. This is also the structure of immunoglobulin G, but little bit different. It is shown a little bit different, and I ask you to copy this figure to your notebook. Please copy this figure to your notebook. This is important because here we see that VH and VL domain form superdomain by hydrophobic interactions, and this superdomain here and here, and there is also superdomain formed by CH1 and Cl, CH1 and Cl, and also by 2CH3. This I have already told you before, but now you can see it simply on the picture. Please tell me why CH2 and CH2 cannot form superdomain together. Think please. And I am answering, I told you about this, because there are carbohydrate components between them, and these carbohydrates do not allow to CH2 domains to interact by hydrophobic interactions, hydrophobic bonds. Let's look at different colors. What is shown by red color? It is understandable. This is antigen binding site here and antigen binding site here. What is shown by yellow color? FAB fragment and second FAB fragment, antigen binding. What is shown by green color? Also it is clear, it is FC fragment. FC part of the molecule of immunoglobulins. This is the hinge region. This line shows disulfide bond, and as this hinge region is the flexible part of the immunoglobulin, you can simply understand that this angle alpha can be different because these FAB parts can move up and down, up and down. And what is also very important, please look at these arrows. They shows the absolute size of the molecule of immunoglobulin. This molecule is very large, I told you about this, 150,000 molecular mass, but the length of FAB fragment is 80 angstrom, when of FC fragment is 70 angstrom. So it is so large molecule that it can be seen by the help of electronic microscopy. Let's go on. My question, what is shown here? Look carefully, and you will, I think, simply understand that this is FAB fragment of immunoglobulin. Blue color shows the part of heavy chain, and red color shows light chain. And yellow is antigen. On this picture you can simply see that the structure of antigen binding site, which is here, is complementary with the structure of antigen. So these two molecules can specifically react. This is the complex of antigen and FAB fragment antigen binding of immunoglobulin. Let's go on. Now we must write two definitions. We must explain what is polyclonal antibodies and what is monoclonal antibodies. On the pictures you see antigen with different antigen molecules on the surface, and by blue and red color you can see it is shown different antibody molecules. Now please write polyclonal antibodies. Polyclonal antibodies are antibodies. Polyclonal antibodies are antibodies that react with the same antigen. Look, all of them are reacting with this yellow antigen, which react with the same antigen but have different primary structure. And here it is shown at least two different molecules of antibodies, red and blue. They are shown by different colors because they have different primary structure. Let's continue our definition. So polyclonal antibodies are antibodies that react with the same antigen but have different primary structure because they were produced by different clones of B lymphocytes. Because they were produced, they were synthesized, they were created, as you want, by different clones of beta lymphocytes. And now you can write the next sentence, that immunoglobulins in our organism appeared as the result of biosynthesis by B lymphocytes. So B lymphocytes is antibody producing cells in our organism. After evolution in our organism, B lymphocytes can be transformed to another type of cells, plasmocytes. So plasmocytes are cells that are also producing large amounts of antibodies. What is clone? We say the different clones of B lymphocytes. What is clone? Clone is the group of the cells which appeared from one cell. So one cell began to divide, and finally it produced a group of the cells which we call clone. So different clones of B lymphocytes produce different antibodies, and we call these antibodies polyclonal antibodies. Now please let's explain what is monoclonal antibody. Right, please. Monoclonal antibodies are antibodies which react with the same antigen. The beginning is the same as in previous definition, which react with the same antigen. Look, with this yellow one also, but all of them have completely equal primary structure. Because they were synthesized by one clone of B lymphocytes. Because they were produced by one clone of B lymphocytes. Because they were produced by one clone of B lymphocytes. Now you can understand why we call monoclonal antibodies monoclonal, because mono, it is one. Monoclonal antibodies are antibodies produced by one clone, and polyclonal antibodies are antibodies that were produced by many clones of B lymphocytes. Normally, when antigen appeared in our organism, different clones of B lymphocytes begin to produce antibodies. And normally the immune answer in our organism is the answer by polyclonal antibodies. But in laboratory, scientific purposes, medical purposes, it is very important to obtain large amounts of monoclonal antibodies of equal structure that can react with the same antigen, with same affinity, and it was a problem for some time for scientists to produce large amounts of monoclonal antibodies. Why? Of course, it is possible to take one lymphocyte from the organism of the animal and wait until this lymphocyte will produce antibody. But the number of such antibodies was very small, because out of the organism, B lymphocyte is living not more than 10 days. So the problem was to force a lymphocyte somehow to live long, to produce the large clone of the cells, which can synthesize a large amount of monoclonal antibodies, which we can use for diagnostics, for treating of diseases, for scientific purposes, and so on. This problem was solved by two scientists. First is Georg Köhler. Sometimes we say George Köhler. He is from Germany, and he worked in Great Britain, and he made his discovery in Great Britain. Georg Köhler. And second one is César Milstein. He was born in Argentine, but also then moved to Great Britain, and they worked in one laboratory together with Köhler. So Milstein and Köhler were working together. This is a different situation if to compare with Edelman and Porter. Edelman and Porter worked in different laboratories, but studied the same immunoglobulin. And Köhler and Milstein were working together in Great Britain. One came from Germany and second came from Argentine. They proposed the method which we call hybridoma technique or hybridoma technology. Please try to copy this figure to your notebook, and I shall slowly explain what we have here. The idea was to make B lymphocyte immortal. What is immortal? Immortal, that can live for a very long time without death, can produce a lot of cells of one clone. Is it possible? Yes, it is possible. Malignant tumor cells are immortal. They can live in vitro, for example, for a very long time if we shall give them food. And the problem that malignant tumors originates from B lymphocytes are producing antibodies which we don't need because we don't know their specificity. The problem that we need to obtain large amounts of monoclonal antibodies with desired specificity. So we need to receive antibodies against antigen which we need. And we need to obtain antibodies with desired specificity. How to do this? Köhler and Milstein solved this problem by hybridization of two cells. Hybridization of malignant tumor cell and hybridization of normal B lymphocyte. So, at first, we have a culture of malignant tumor cells. These cells are immortal because they are malignant. And now we need to receive second cell, the B lymphocyte, which is producing antibodies with desired specificity. How to obtain it? For this, Köhler and Milstein used mice, this white mouse. Look, it is shown that it is we have here immunization. By the syringe we inject antigen, and later this mouse will produce antibodies against injected antigen. Then we take B lymphocyte from this mouse, and these B lymphocytes we put in the same test tube with malignant tumor cells, and then we forced junction or fusion of these cells. Finally, we have, as we used a lot of cells, we used many hybrids, and also we have some B cells which were not fused, and also some malignant tumor cells which were not fused, which after this fusion still living separately. So finally we have a mixture of different cells, hybrids and non-hybrids. Now we must find the cell which is hybrid cell, which is immortal as a myeloma cell and which is producing antibodies, monoclonal antibodies against antigen which we injected to the mice. How to do this? As I told you, we simply can wait 10 days, and after this normal B cells which did not produce hybrids will die themselves. But now we must also somehow kill non-hybridized myeloma cells, malignant cells. For this we use special HAT selective media in which malignant tumor cells are dying. Finally, we shall receive in our test tubes only hybrids, because they will not die as malignant tumor cells in HAT selective media, and they will not die as normal B cell after 10 days. But among these hybrids, which we now have in many test tubes, there are hybrids that produce antibodies and hybrids that not produce antibodies. Now we must find test tubes with cells which are producing antibodies. How to do this? Here you see on the next figure, you have new test tubes in which we have only hybridoma clones. Then we are doing procedure which we call cloning. What is cloning? we take, for example, from one of these test tubes, very small amount of cells, for example, only 30 cells, and pour the solution of these cells to 96 test tubes. So only 30 cells to 96 test tubes. Finally, we can sure that in each test tube will be not more than one cell. Then we are waiting for the growth of these cells and do this procedure second time, recloning, to be sure that in each test tube we have only one of clones of hybridoma cells. Hybridoma is the hybrid tumor. Oma is tumor, hybrid is hybrid. So finally, in these test tubes, in each of these test tubes, we have only one clone of hybridoma antibody producing or antibody not producing cells. Now we must be sure what clones are producing cells, what clones are not producing cells. For this we use ELISA method, which I shall show you on the next figure. But let's decide that finally we obtain a test tube in which we have clone of hybridoma or producing antibodies which desired specificity. What do we do with these cells? We have two possibilities. First possibility is inject these tumor cells to the mouse, to the abdominal cavity of mouse, or to use special bottles with media for producing a large amount of such cells. And these cells are producing large amount of antibodies. Now look at this figure. It shows the ELISA method, enzyme linked immunoassay. ELISA, enzyme linked immunoassay. So we take many test tubes. On the bottom of the tubes we fix molecule of antigen. This was the antigen, look on this figure, which we injected to the mouse. This is antigen against which we need to obtain antibodies. Let's look what will be further. Then we use the second solution, which we take, let's look here once more, from these test tubes from media. And if they one moment, if they have antibodies, here they are shown with the red color. If they have monoclonal antibodies, these monoclonal antibodies will react with our antigen. Then we must understand that in our test tube, we have this interaction. The next stage, we pour solution of antibodies against mice antibodies, and they specifically react. Look here, and these antibodies are marked with special enzyme, for example peroxidase, and this peroxidase will change the color of stain that we also add to this test tube. So we have system with few components, antigen on the bottom, then a monoclonal antibody, then antibody against monoclonal antibody together with enzyme. Then we add stain, and this enzyme change the color of the solution. Finally, in the test tube, in the test tube we can see the change of the color, and it means that in this test tube we have monoclonal antibodies which we need to obtain. Let's go on. For these investigations we use special mice. This is a line of mice. In our experiments we used BALB line of mice. All of them have completely same antigens, and this is necessary to use hybridomas to inject from one mice to another and to receive hybridoma cells, hybridoma clones without problems, without force, battle of the immune system of these mice against these hybridoma cells. I want to tell you that Köhler and Milstein, about which I told you, published their investigation devoted to creation of hybridoma technique in 1975. And in 1984 they received the Nobel Prize. 1975 publication, 1984 Nobel Prize. And in 1986, 2 years later, we created hybridoma laboratory in our department, in the Department of Biochemistry of our medical university, in our medical academy. And this laboratory is still working, and I want to show you some photos from our laboratory. These are, as I told you, our mice that we use for experiments. This is the heart of the laboratory. This is CO2 incubator. Inside of this cabinet there are conditions same as inside of the body of the mouse. What does it mean? Inside we have 100% humidity, then 5% CO2 concentration, and temperature 37 degrees. So when we make different procedures with cells, with lymphocytes or myeloma cells or hybridoma cells, we can put our bottles inside of this cabinet, and cells will live inside without problems. Next, this is showing that the temperature is 37, 36.9, humidity and other conditions inside. Then all procedures must be done in special on the special table. The secret of this table that here there is a very strong ventilator that move the air stream through special filters, and finally when scientist is sitting here, the sterile air is coming to his face, and as a result there is no contamination of sterile medium, sterile cell cultures by different bacteria which can come from the scientist. This is the plate consisting of 96 small test tubes in which we make all our procedures. You can simply calculate that we have 96 small test tubes, because this row is 8 and this row is 12. All procedures must be done very quickly, because we do them in sterile conditions, and for this we use special pipettes with sterile tips. So you see that this pipette has 12 tips, and so we are doing 1, 2, 3, 4, 5, 6, 7, 8 movements, and all 96 test tubes are finished. So it is very quickly. This is the mouse inside of which hybridoma cells are living. So these hybridoma cells forced the mouse to produce acidic fluid, and this acidic fluid in the abdomen of the mouse has large concentration of monoclonal antibodies. So we can use syringe and take some such fluid and receive monoclonal antibodies from such mouse. One mouse can produce up to 10 milliliters of acidic fluid, and the concentration of antibodies in each milliliter is approximately 10 milligrams. So one mouse can produce 100 milligrams of monoclonal antibodies. If we will take many mice, we can receive much volume of this fluid and large amount of antibodies. And now, at the end of our lecture, I want to tell you about some applications of monoclonal antibodies. For example, on this picture you can see affinity chromatography. If we use monoclonal antibodies that can specifically interact with one of the molecules, such molecule is shown here by blue color, we can use such antibodies to divide different molecules. For example, we have mixture of blue and black molecules, then we pour this mixture to the test tube with monoclonal antibodies. Blue molecules will fix and black not. And finally we can divide them and receive two maximums. One is black, one is blue. But another example is much more interesting. Please write the word immunotoxin. It is written here, look. Immunotoxin. What is immunotoxin? Let's write a definition. Immunotoxin is the molecule of immunoglobulin with desired specificity combined with a toxic molecule. Combined with toxic molecule, with poison. If we inject such a molecule to the patient, these monoclonal antibodies will find, for example, malignant tumor cells in the body of the patient, and this poison will kill them. So in this case, we can decrease the number of the drug which we use against the malignant tumor, because it is coming only to the pathological cells. How to obtain this immunotoxin? We take mouse and apply hybridoma technology. The figure which you already had in your notebook, now it shows only with this small arrow. We made all these procedures, and we received monoclonal antibodies. Then we take a toxic molecule, toxin, poison from poisonous plants or from poisonous bacteria, and then combine them together. One of the most strong toxins in the world is diphtheritic toxin, which can be produced by special bacteria. But this diphtheritic toxin consists of two parts. One interacts with tissues of the organism. Second one is the real poison. We must divide them because we need only poisonous part, which we shall combine with our monoclonal antibody. So we divide this molecule into two parts, A chain, B chain, and then look, A chain, B chain we took away, and A chain we chemically combine with Fc fragment of monoclonal antibody. Finally, we have immunotoxin. Then, how to use it? If we inject such immunotoxin, molecule of immunoglobulin with the molecule of drug into the human body, this antibody will find special antigens. You remember these antigens we used for immunization of the mouse in hybridoma technology, in the right upper corner on our picture. And for example, these antibodies can react with specific antigens of tumor cells. So these immunotoxins will find tumor cells in the body of the patient, interact with them, then goes inside of the cell, and this toxin kill the pathological cell. This is the complete scheme. I recommend you to copy it to your notebook. So let's explain step by step. For example, we have a patient with a malignant tumor. This is the cell of malignant tumor from the patient which we expect to treat. On the surface of the malignant tumor cell there are different antigens. For example, these black triangles shows the specific tumor antigens. Then we take these cells from the organism of the patient and use them for immunization. So look, there are tumor cells inside of this syringe. Then mouse will produce antibodies against tumor cells, against specific antigens of tumor cells, and we use here hybridoma technology, and finally we receive monoclonal antibodies in large amounts that can specifically react with these black antigens on the surface of the tumor cells. Then we take a poisonous bacteria or poisonous plant, purificate toxin, and chemically combine immunoglobulin monoclonal antibody with this toxin. And finally we are receiving immunotoxin. Then we inject this immunotoxin to the body of the patient. If a normal cell will be on the way of this immunotoxin, there will be no interaction because there are no these black pathological antigens of the surface of normal cell, and normal cell will survive. But when immunotoxin meet tumor cells with specific pathological antigens, it will immediately react with this cell, and finally cell will be killed. This is the dead malignant tumor cell. So once more, patient with tumor, we use tumor cells for immunizations. For immunization, we receive by hybridoma technique specific antibodies. Then we combine these antibodies with the drug, with the poison, which must kill malignant tumor cells. Then we inject this drug to the body of the patient. Normal cells will still live, and malignant tumor cells will be killed. How killed? You see on this picture. They came inside of the cell, and the drug kills the malignant tumor cell. If we shall take larger magnification, we can see it here. I want to say that now we have many different drugs on the base of monoclonal antibodies, which are very effective against different types of tumors. Sometimes even we don't need to combine monoclonal antibody with the poison, because after interaction of the tumor cell with antibody, complement system will kill this tumor cell. There are many different types of such monoclonal antibodies, from which scientists can take only antigen binding fragment, or even only the variable and variable domain of heavy chain, a variable domain of light chain. Or even sometimes they can take only antigen binding site and include it to the human immunoglobulin. This is very important, because if antibody from mouse will appear in the organism of the human being, it will be an immune response against the foreign antibody. So if the antibody is human and only antigen binding site was taken from the monoclonal antibody, there will be no real immune response until this antibody will fix on the surface of tumor cells. So now we have great progress in this technique, and I hope that in some decades very many malignant tumors can be treated by different types of the molecules made on the base of monoclonal antibodies. If you have no questions, our mice are saying you goodbye. Best wishes. I hope we shall meet at the next lectures. Goodbye also from me.